Meiosis is a type of cell division that makes gametes from cells in the gonads. It produces haploid gametes from diploid body cells. It starts out with the duplication of chromosomes and is followed by 2 rounds of cell division. To put it in easier terms, the chromosomes are first doubled, then divided in half, and then divided in half again. There are 2 phases of meiosis, but before they can take place, interphase must occur. During interphase, the cell is growing, carrying out its cell processes, and replicating its DNA. During DNA replication, the initial 46 chromosomes that the cell starts out with is doubled to make 92 chromatids. First step of meiosis is prophase 1. During prophase, the chromosomes condense and line up by homologous pairs and exchange corresponding segments, which essentially means they're like swapping information with each other. The next step of meiosis 1 is metaphase 1. In metaphase 1, the homologous pairs align in the center of the cell. Once metaphase 1 is complete, anaphase 1 can begin. The homologous pairs of chromosomes separate from one member of each pair and head to the opposite side of the cell. The final stages of meiosis 1 are telophase 1 and cytokinesis. This is where the nucleus begins to reform and the cell divides into 2 haploid daughter cells. Phase 2 of meiosis can begin. The first step of meiosis 2 is prophase 2. This is where the chromosomes condense in the 2 cells that were produced. Next comes metaphase 2, and then just like in metaphase 1, the chromosomes line up in a straight line in the center of the cell. Once metaphase occurs, anaphase 2 begins. The sister chromatids split apart, with one copy distributed to each side of the cell. The final stage of meiosis is telophase 2 and cytokinesis. Each cell splits by cytokinesis, making 4 offspring cells, each with half the number of chromosomes as the starting cell. The nuclear envelopes reform and the chromosomes uncondense. At the end of meiosis, there are 23 chromosomes and 23 chromatids.